Good morning, everyone. Myself, Dr. Sakshi Bharadwaj, on behalf of Altum Technologies, I welcome you all in one more very interesting session of our Women's Day special webinar series. This session is entitled as Ayurvedic, a novel tool in Ayurveda research. I'm thankful to all of you that you could have joined us today. Do you know which medicine science is called as ancient system of life and also oldest surviving medical system in the world? Yes, you are correct. It's Ayurveda. It's a science of life, and its name itself is like Ayur is as life and Veda as science. It is also regarded as an antiquated therapeutic science with about 5,000 years of history that enhance longevity. Hence, it is often called as mother of all healings. The rishis or the sages of ancient India had a deep understanding of creation and maintenance, which leads to the development of it along with their desire to live happy lives. The foundational principle of Ayurvedic system are universally applicable, making them easily adaptable to the new situations and the centuries. The philosophy of Ayurveda teaches actually that a person's mental, emotional, spiritual being are all interconnected, and any kind of imbalance in between these results in health disorder and diseases. According to Ayurveda, there are three fundamental forms of energy. You might have heard also, those are vata, kapha, and pitta, right? So basically, the vata is the energy of movement, whereas pitta is the energy of digestion or the metabolism, and kapha is the energy of lubrication and structure of all of your cell and those kind of things. Presently, medical research has experienced a paradigmic shift from in vivo to in silico experimentations, a development that relies mostly upon the bioinformatic tools. Hence, we have Ayurinformatics, the need of today. To explain the bioinformatics in Ayurveda and how we can take the advantage of our ancient science merging with the modern tools, we have our speaker of the day, Dr. Pallavi S. Jamadagni. She is a research officer, Pharmacology, Regional Ayurvedic Institute for Fundamental Research, CCRAS, Ministry of Ayush, Government of India, Pune. She has done her undergraduation and master in veterinary science. She held various positions, including study director at Intox Private Limited in Pune, lecturer at Bombay Veterinary College, Bombay, veterinary consultant at Department of Clinical Pharmacology at Nair Medical College in Bombay, senior research fellow at CCRAS headquarters in New Delhi, and research officer pharmacology at NRI-ADD in Kolkata. She has 15 research articles to her credentials. She has 11 completed IMR research products as well. Now, without taking much time, I would like to hand over the platform to our expert. Up to you, ma'am.
Yes, very good morning to all of you. So, first of all, I'd like to thank Sakshi for this nice introduction. And the way she was speaking on Ayurveda is really wonderful. She appreciated Ayurveda, and it was very nice introduction of Ayurveda. And again, Ultim Technologies, I'm very much thankful because they have provided us BioVia Drug Discovery Studio, which is very much user-friendly tool we are using, and that is why we could establish this department of Ayur-informatics, or we can work on the principles of Ayurveda with the help of the Drug Discovery Studio. So, with this, let us start with our topic today. It is Ayurvedic, a novel tool in Ayurveda research. So, as we discussed, Ayurveda is a complex system and bioinformatics is a simplified system. Why Ayurveda is a complex system? Because it is not like allopathic drug that if you are having fever they'll give you paracetamol. If you are suffering from diabetes, then they'll count the sugar, blood sugar level, and then they'll give you some uh, anti-diabetic drug. And it is not like that in Ayurveda will see how the person is, what is the body disposition of the person, and then they'll give you medicine. That is the holistic approach of a medicine, and that is why it is a complex system. Whereas bioinformatics has tried to simplify all the complex mechanisms which are occurring in the biology, and then bioinformatics helps all other systems to describe in a more measurable or quantifiable way. So this is Ayurveda is an pretty old system, and bioinformatics is a little bit new system, and integration of both of these can be summarized in Ayurinformatics, wherein we will try, or scientists are trying to integrate the knowledge of Ayurveda with bioinformatics and develop some translational value research. So, what are the characteristics of Ayurveda? Ayurveda is basically, as Sakshi has told, it is based on three dosha, Vata, Pitta, and Kapha. Whole the population is classified into three: Vata Prakriti, Pitta Prakriti, and Kapha Prakriti. And then, based on their symptoms, the medicine is given. Now, formulations in Ayurveda, they contain natural resources. The formulations are from natural resources. So, one formulation may contain various plants, and that to specific parts of the plants. And then these plants of the parts of the plants may contain various phytochemicals, which cause the bring about the action of the drug. So various natural resources are used, and their combination helps to holistic treatment of the disease. And also Ayurveda uses toxic plants which are used after purification. It uses heavy metals in bhasma preparations, and these both toxic plants and heavy metals, they are used after purification. So purification system is also defined in Ayurveda, wherein in plant purification they use gomutra and godugdha, and in heavy metal preparation repeated heating and cooling is performed, and that is why there is a decrease in the particle size, or increase in the potency, increase in the bioavailability, decrease in the toxicity. Such procedures are adopted. The purification process is called as shodhana, and that is why it is the only system which uses toxic plants for the treatment, and also heavy metals for the treatment of the disease. Then again, it is a tailor-made medicine, as we have discussed. Vata, Pitta, Kapha, Prakriti is diagnosed, and based on that, what medicine is to be given, what will be the effect of medicine, if what the dosh, Vata, Pitta, Kapha, the medicine is supposed to cure. Based on that, the medicine is prescribed. That is why it is called as tailor-made medicine. And again, holistic approach, not only the physique of the person, but the environment and the symptoms, and based on all other factors affecting the disease, the medicine is prescribed. So some medicines are prescribed as a therapeutic, and some they will support the main therapeutic agent. So likewise are the characteristics of Ayurveda, which are very important from bioinformatics point of view. So in bioinformatics, we have seen there are software tools which are used to understand the biological data. Bioinformatics analyzes the relationship among various elements and illustrates the biochemical reaction between metabolic substrates and products, and takes into account huge databases generated through research. So bioinformatics is quite understanding large data and interpreting some conclusive interpretation, so that we can come to know what is the mechanism of action, or what is the underlying proteins coming into the picture, what are the genetic changes. This all bioinformatics will help us to understand. Now, what is the gap in research? As we have seen, the Ayurveda takes into account Prakriti, that is Vata, Pitta, Kapha, Sapta Dhatus, they are present in the body and their interplay, and Panchamahabhuta, that is uh, air, water, and space, like that. So there is much integration of these Prakriti, Saptadhatus, Panchamahabhutas, and other factors which are there in Ayurveda. But some of the factors are immeasurable. Like you cannot measure the effect of Panchamahabhuta, or you cannot measure the Panchamahabhuta. And there is need to generate an evidence that can be seen as genetic change, protein expression, or other molecular parameters that can be quantified. So the quantifiable parameters need to be expressed, which are related to these immeasurable things which are there in Ayurveda, and scientists are trying to establish a mathematical correlation between these factors. So Ayurveda is basically application of bioinformatics to Ayurveda, and as we have seen in bioinformatics, there is in-silico drug discovery and network pharmacology. In-silico drug discovery will reveal the active phytochemicals, and network pharmacology will understand the pharmacological mechanism of active phytochemicals. And this both in silico drug discovery and network pharmacology will definitely have some conclusive effect on Ayurveda, and that is why Ayurinformatics needs application of bioinformatics to Ayurveda. So, how can bioinformatics help Ayurveda? Ayurveda classifies the whole human population into three major constitutions as Vata, Pitta, and Kapha, and their possible combinations. So, at a time, a person may be of Vata Prakriti and some Pitta dominance, and likewise for Pitta Prakriti and Vata dominance, and that is why the interplay of these causes the disease. And their homologous relation to human genetic structures need to be studied for validation. So scientists have already started working on some single genes, and then their interrelation with the Vat, Pitta, and Kapha, and the many things are under validation. And if these things are these relations about to the human genetic structure, if they are validated, this hypothesis will have far-reaching implication for pharmacogenomics, modern genetics, and human health and Ayurveda. So it is very important to understand the interplay of the basic Vata, Pitta, and Kapha, which are there in the body. Now, involvement of what is Vata, Pitta, and Kapha. Vata is involved in movement, which includes muscles contraction, respiratory and reproduction, and circulatory system. So it is majorly a function of Vata. And Pitta is involved in metabolism and digestion, and also responsible for hormonal and enzymatic activity. And involved the kapha is involved in formation of structural basis and lubrication of the body. So these three are involved in basic functions of the body, and that is why it is important to understand their interrelation, which can be done with the help of bioinformatics. So the interplay of the three dosha plays integral part in regulating different functions of human body. So when one dosha is affected, so will be the other, and therefore the balanced state between the three dosha equals health, and whereas imbalanced state signifies a disease state. So it is very important to understand the balanced state and imbalanced state, and then we will be able to understand the basic underlying mechanism of a disease. So, how the Ayurveda treatment is done? Ayurvedic treatment includes, first of all, first stage includes internal purification process. Then they give special diet, and then herbal remedies, and then meditation, yoga, and massage therapy. So, all these processes have some relation to the proteomics or metabolomics, and then they bring about the change in the proteome or metabolome or epigenetics, and then which can come under the umbrella of bioinformatics and can help us to understand the effect of this diet or this medication or the other yoga therapy and massage therapy. So bioinformatics can be applied in these fields. What are the recent developments? Recently, people have started working or taking help of information technology in Ayurveda. For example, some have developed vision based Prakriti Pariksha based on the hair, eye, nose, lips, skin color, etc. And they process the image with the help of software and uh, give interpretation which Prakriti the person is. Again, Nadi Pariksha and its relation to the pulse. People have started establishing relation with the help of information technology. Prakriti analysis based on a questionnaire. Already CCRS has developed Prakriti analysis portal in which they ask you some questions, you have to answer, and then finally you will get analysis of Prakriti, which is your major Prakriti and what is the what are the other prakritis involved. So major and minor prakritis, they give you an interpretation. Then again, there are many electronic health record systems which are available. Ayusoft software, some might have seen, which is developed by CDAC. So in which there you have to insert or you have to input the history of the patient, and then based on the condition of the patient, its prakriti dominance, the advice for treatment is given and the record is maintained. So various tools people have developed for electronic health records, special reference to Ayurveda. And now what statistics says is the quantitative measure of tridosha is obtained by applying and heuristic algorithm approach to the exhaustive list of qualitative features that Ayurvedic doctors they commonly used. And this is a knowledge-based concept of worth coefficient and fuzzy multi-attribute decision functions which are used with the help of regression modeling. So heuristic approach is the approach in which you will get a prediction of the Prakriti based on the qualitative features which are given by the Ayurvedic doctors, and then the interpretation can be done which Prakriti is dominant. And here some coefficients are there which are given a special attribute, and then the interrelation between the prakritis is obtained. So heuristic approach gives you a near to true or near to real diagnostic feature. And although this approach has to be validated, and if the approach is or heuristic approach algorithm is more than 90% valid, then it can be used for larger population. So, in this way, bioinformatics can help Ayurveda. There are certain examples in which Prakriti analysis was done. 300 patients with coronary artery disease, more than 25 years were studied, and Prakriti assessment was done with the help of ISOF software. And biochemical parameters, inflammatory markers like HSCRP, TNF alpha, and IL 6, and insulin resistance were measured. And it was interpreted that there is a strong relation between risk factors, that is, diabetes, hypertension, or dyslipidemia, insulin resistance, and inflammatory markers with Vata, Kapha, and Pitta Prakriti. So, such studies are already done, which needs to the database development, which can help in the development of the database. Again, is there any correlation between genes and prakriti? So 76 subjects were evaluated for their prakriti and human leukocyte antigen, and the various types of it, HLA, DRB1 types, and genomic DNA was extracted with the help of PCR. This gene was evaluated, and it was found that there is a correlation between HLA type and Prakriti type. So, complete absence of HLA1, HLA-DRB1, 2 allele in the Vata type, and HLA-DRB1 13, number 13 in the Kapha type is significant. HLA-DRB1 10 has a higher allele frequency in the kapha type and in the pitta and vata, less than in pitta and vata prakriti. So such genetic correlation is already established between the genes and the prakriti. So this needs to be considered for application in bioinformatics. Also, Ayurveda assigns Prakriti not only to human but also to the environment and food, and it makes specific mention of adaptation as well as dietary and lifestyle recommendations based on one's Prakriti for achieving the healthy balance. A study has shown that the Pitta constitution is more protected at high altitude, which would be consistent with the Ayurvedic school of thought that considers mountains mainly as Kapha and Vata dominant region. So again, it assigns Prakriti to the environment, and Kapha and Vata dominance is there in the mountains, and that is why Pitta constitution is more protected in the mountainous area. So the disease prevalence will be Kapha and Vata related diseases. Expression of genetic analysis of healthy individual phenotype using principles of Ayurveda could uncover genetic variation that are associated with adaptation to the external environment and susceptibility to the diseases. And there are various concepts in Ayurveda. Concept of rasa is very important. Rasa is the taste of substance, that is the foremost tool in Ayurveda to assess and determine the pharmacological property and action of the substance. The similarity in rasa is said to signify similar structure and consequently similar pharmacological behavior. So, two plant material or two drugs, if they are having same rasa, then it is postulated that their structure may be similar and then their pharmacological behavior may be similar. So one can be used as an alternative to another. So pharmacological behavior of the drug is basically these four parameters are considered while considering the pharmacological behavior of the drug: Rasa, Guna, Virya, and Vipaka. And Rasa refers to complex totality of experience arising from all the perceptory interactions of the material, like mouth and nasal passage, taste buds, and olfactory receptors. So, rasa is what body understands the drug is. For example, longum is madura in fresh condition. It is considered as sweet in fresh condition, and in dry form, it is considered katu or bitter, and that is why fresh longum is considered heavier to digest. That is guru to digest, and dry longum is easy to digest, and that is why the recommendation is given whether to use fresh condition longum or to use dry longum. Then, by knowing the rasa of substance, one can decipher the potent pharmacological properties and actions of the substance. For example, if the substance is madhura in rasa, then it is understood to have prithvi and jala mahabhuta as in active state. The substance is heavy in digestion, sluggishness, it imparts sluggishness to the body, anactus in nature, and facilitates anabolism, causing compactness of the body and moistening the body channel when we say something is madhura. And based on these properties, a substance can be clinically used as a general or specific tonic or purgation. So, various uses of the drug or a substance can be considered based on its rasa, and whether it is to be given as a tonic or to be utilized as purgation or to be utilized as a supportive therapy or to increase the absorption of the drug based on the rasa prakruti, rasa of the substance, the decision is taken. Now, virya is again potency or active component of the drug. It is divided into three: mridu virya, madhyama virya, and tikshna virya. It is related to action of the drug, how if the drug is taken in, how it will act. So it is the energy content in the drug, it is related to active principles of the drug, and uh, it is related to the generation of action, how the drug will act when it will enter into the body. And in pharmacological terms, it is related to potency of the drug. So, Ayurveda defines Virya in Mridu or Madhyam or Tikshna form, and then the decision for giving medicine is taken. Vipaka is specific transformation or effect of drug after digestion. So, what body does to the substance is comes under the Vipaka. It is one kind of pharmacokinetic, we can say. The process of metabolism is divided into two main components, that is Avasthapaka and Vipaka. Avasthapaka is the general process of catabolism needed for food and medicine ingested orally. It involves three stages, that is Madhura, Amla, and Katu. When it is Madhura, it is difficult to digest, and it is converted to Katu, that is easy to absorb and digest. So, Vipaka mainly starts in the stomach and duodenum and is responsible for ultimate effect of the food or medicine ingested. Vipaka is defined as a transformation occurring in the nutrient fluid after the action of Jamadagni, that is the intestinal or juices coming from the stomach. So Vipaka is the change in the uh, pharmacological substance which happens or which occurs in the stomach. Again, Guna is again it defines the drug qualities of the substance, whether it is heavy or light, hot potency, cold potency, oiliness, non-oiliness, dullness, sharpness, immobility. So various gunas are attributed to the drug, and it is again related to the Panchamahabhutas, and based on the guna, the choice of the drug is done. So if the requirement is to increase the digestion, then the gunas of lightness guna is selected, or likewise the decision is taken in Ayurveda. So in Ayurinformatics, we need to consider all these pharmacological properties, that is Rasa, Guna, Virya, and Vipaka of the drug, and that is why Ayurinformatics needs to consider basic principles of Ayurveda. We have seen various changes in Ayurinformatics, that is before COVID, there were very less publications related to air informatics, and post COVID, there was increase in publications related to air informatics. And the main reason is that there is a shift to the from the modern medicine to the basic Ayurvedic medicine, and people started believing that the phytochemicals or phytoconstituents or drugs based on plants are also helpful in improving the immunity. So, screening of phytochemicals for specific activity is one application of Ayurvedic. So, in this, again, we can find out the underlying mechanism of the drug. Ayurvedic formulations, which contains various plant parts. They contain a lot of phytochemicals, and then those phytochemicals, they have different mechanisms and their target disease. So this will help us to find out the underlying mechanism of the drug. For example, immunomodulatory activity of Vidania somnifera was studied, and then bioactives were taken. A large list of active compounds or phytochemicals was considered, and their respective targets were found, and a network was built between the bioactives and targets, and then their internal pathways was studied, and protein interaction network was studied, and then it was postulated that Vidania somnifera has got a large immunomodulatory network, and definitely few of the phytoconstituents of which are present in the Vidania somnifera, they have immunomodulatory properties. Again, neuroprotective activity of Saraswata Rishta was studied, which contains 15 ingredients and 534 phytochemicals. And then it was studied for dual leucine zipper kinase, which plays a role in neurodegenerative conditions. And docking was performed. Top 10 good binding phytochemicals were considered, and then their absorption, distribution, metabolism, excretion, toxicity studies, prediction was done based on the databases. And then it was found that these four phytoconstituents, these four plant material, are basically responsible for the neuroprotective activity of the Saraswata. And again, anti-diabetic activity of Kathakadi Kashaya was studied, in which again phytochemical data mining was done, and its molecular docking was done. Based on the simulation, the stability of the protein and drug binding was studied. Network pharmacology was done, and it was found that this out of these phytoconstituents or plant material, most active are curcumin or strychnine, which bring about the antidiabetic activity of Kathakadi Kashaya. So around 37 proteins were identified which are related to diabetes and which are affected by the ingredients of Nishakathakadi Kashaya. So this is the network in which you can see the yellow nodes are the compounds present and the pink nodes are the protein targets. Much protein targets are related to the uh, ingredients of the Nishakathakadi Kashaya, and out of that, these uh, yellow, these blue and orange. So, blue are the phytoconstituents, and orange are the diabetes related proteins. So, the interrelation between the diabetes related proteins and the phytoconstituents was established, and it was predicted or postulated that this kashaya has got good anti-diabetic activity. Again, triphala in angiogenesis was studied, various proteins were considered, and then the phytocompounds were isolated, and based on the prediction efficacy, it was suggested that the triphala has got good activity in angiogenesis. Again, it was studied for anti-inflammatory properties, and also triphala has got good anti-inflammatory property and anti-angiogenic prediction. So, based on the prediction efficacy, it can be studied whether the drug is efficacious or which part or which phytoconstituent of the uh, drug is the most active. Then Chandanasava is again studied in chronic kidney disease, and after this network pharmacology, it was found that piperine and melatonin, which are present in Chandanasava, plays an active role in reversing the chronic kidney disease related complications. So, such studies were performed in which the Ayurvedic classical formulations were considered, their active phytoconstituents were taken, and their relation to the proteins was established. It was confirmed by the docking, and then it was the most active phytoconstituent was determined, and the plant containing that active phytoconstituent was attributed, and this will help to establish the quantitative measure that if certain phytoconstituent is more in the formulation, that formulation is very much effective in certain disease. So based on this, we can easily establish the correlation. So this is a list of various databases which are helpful in generation of the network in Ayurinformatics. And prediction efficacy is the calculation of prediction efficacy of a compound will give you a score, that is the docking score. Uh, it is a summation of the docking score with various proteins. So P is the set of proteins related to disease, and X is the docking score between these compounds and the protein. So a certain compound can be given prediction efficacy in certain disease if it is docked against the proteins which are available for which are responsible for that disease, and then the prediction efficacy of that compound can be drawn. And same way, prediction efficacy of that plant in certain disease can be calculated. That is summation of the score of that compound, prediction efficacy of that compound, and that can be attributed to the plant. So this can help in establishing a good relation between Ayurveda and informatics, and this will help in prediction of the mechanism of action of the Ayurvedic drug. So the scope for bioinformatics is genetic basis of Prakriti analysis, which we have already discussed. Network-based mechanism of action prediction of some classical formulation. It is very much, we can say, demanded area, because people are taking network-based people are taking formulation. They are finding out the phytoconstituents, and then they are establishing the network, and then they're predicting the mechanism of action of that drug. And this will help in, this is again having translational value in Ayurveda. Then toxicity prediction of Ayurvedic drugs based on phytochemical constituents is essential, because Ayurvedic drugs are mostly blamed for containing toxicities. But if we see the prediction, we will be able to give a good data of the toxicity of that Ayurvedic formulation. Then prediction of herb drug interaction, whether the if the herb is interacting with the modern drug, then we can predict this based on the Ayurvedic tools. And translation of ancient medicine into modern medicine, again, Ayurinformatics has got good scope in this area. And application of knowledge for the new drug development. Some formulations may be repurposed based on their predictions uh, to be efficacious in certain diseases. So, again, this knowledge can be applied for new drug development. So, with this, we stop here. Thank you. We are open for the questions, and this was generally a basic idea of use of Ayurvedic informatic tools in the field of Ayurveda.
Thank you so much, ma'am. It was very knowledgeable session, as we always heard about that simple living, high thinking. So I think nowadays we youngsters are mostly in the gyms. So as we can adopt that, the lifestyle of our ancient people, like we have to have this good food for the health, and meditation, yoga, and little exercise that could be very great for keeping our health, at for us healthy as usual, as you told and you have explained with your presentation. I think we have some questions. Let me take some questions here. So is it possible to understand the Virya and Vipaka of Ayurvedic drugs via bioinformatic tools?
Yes, actually, what we are trying to establish relation between this Vira and Vipaka, and the proteins which they are affecting with the if a certain plant is having good virya, or having a specific vipaka, then what proteins are expressed, and then based on that we can develop a network. So we can study, we can understand the virya and vipaka based on the Ayurvedic or bioinformatics tools.
Okay, thank you, mom. Next question. How do you identify the target proteins?
Okay, so for identification of target proteins, we know there are various databases available, or we have to perform some studies in lab and find out which protein is upregulated, which protein is downregulated, and based on that, we have to find out the target protein. So we can either do literature search, or perform some experiments, or find out based on the available, which is available in the databases. So if you put proteins related to diabetes or proteins related to Alzheimer's, you can find many proteins. So you have to decide which protein you have to take, and whether it is a human protein or an animal protein. Based on that, we can find out the target protein.
Okay, thank you, ma'am. Next question. How we can predict the interaction of herbs with the targets?
Yes, again, we can do some in-silico docking studies. We can decide some target proteins and perform the docking studies. If the docking score is good, then we can find out the interaction. Again, we have to go for simulation and find out the whether the interaction is stable or not. So we can predict the interaction of herbs and target.
Okay, so I would like to add on this one because as we have the Discovery Studio where you can study the pharmacokinetics, even the pharmacodynamic studies.
Yes.
Even the Discovery Studio is having very unique feature of TopCat where you can study the toxicological properties of each and every component, whether it is from the phytoconstituents or any artificially one or the synthesized one or the ligand molecule, that the small component. Then addition to that, we have the different docking methods in the Biodiscovery Studio. Like we have C-Dogger to check the interaction in between your targeted protein and the small molecules. Then of course, from very curiosity from my side, actually, ma'am, I have this thing that Ayurvedic medication mostly is having a long time of span to to treat a particular disease condition. So if we can have that addition from the bioinformatic tools, as we have many medication which are coming up, or we have like antituberculosis even having the same kind of this thing, like those antitubercular drugs are taking long time to treat the particular disease. So here also we can we can take the advantage of these bioinformatic tools where you can reduce this lifespan to get one potent component actually, so that it will be helpful to reduce that particular time duration for treatment of any kind of any disease. So yes, of course we have that addition. Can we have? Yes, ma'am, are
Yeah,
you?
we can find out the most which is, I mean, as you said, tuberculosis or what, the diseases which are difficult to treat by modern medicine or which take a longer duration. Ayurveda can definitely help in improving the quality of life of that patient. One thing. Second thing is, some of the Ayurvedic medications, they also help modern medications increase the potency or increase the bioavailability of the modern medications. And thirdly, although Ayurvedic treatments are very slow, then but they have some definitive effect. As we have seen, it is a holistic approach. So first treatment line will be based on the purification, or they will first of all, they'll correct the prakriti. If the vata dosha or doshas, vata or pitta or kapha doshas, they'll correct, and half of the cure is done there itself, and then they'll give some herbal remedies. So, following the Ayurvedic methodology, a disease can be cured very easily. And how bioinformatics can help is, bioinformatics can help in deciding the which drug to be given, or to understand the basic underlying mechanism of the drug.
Yes. So can we have more questions? Okay. Can you elaborate on how to convert the Ayurvedic formulations related complex information into suitable information that can be used in informatic methods?
Yes, Ayurvedic formulations. Actually, people are working on that, and um, what we can say, Ayurvedic formulations, they contain many phytoconstituents, and then uh, many plant materials, and then their phytochemicals. So large databases are already available. You can see Impact, you can see this Duke's database. Various databases are available, and you can find out the phytoconstituents present into the plant material, and based on that you can use that data for using bioinformatics tools.
Yes, thank you, ma'am. How to identify active phytochemical and target protein in compound drug.
How to identify active phytochemical is the one which is based on the network pharmacology, as we have discussed. We can find out the phytochemical which is causing that effect. Based on the network pharmacology and docking studies, we can find out the phytochemical which is present, whether that phytochemical is causing specific effect. That we can find out. Target protein you have to search, and you have to decide which protein you are going to target.
Yes, actually, that this question is most or the usually asked question: how to target a protein component
Yes,
or a macromolecule, because that is the most challenging part of bioinformatics. Actually,
yes,
if you have
yes,
identified the protein target correctly, then your half work is done there. So you can take your phytochemicals or whatever the small molecules for the further docking studies or the simulations kind of in studies, right? So, do we okay? Ma'am, can you please explain more about network-based mechanism?
okay. So network based mechanism is, first of all we have to find out the bioactives, then their targets. See, this is all based on the databases. I think Sakshi can also elaborate more. I'll give some baseline information. Um, so we have to find out the bioactive components or chemicals which are present. We have to find out the targets of that chemicals, and then in which pathway these chemicals or these proteins are involved, and based on that we can construct a network. Again, we have to take help of some bioinformatic tools like cytoscape and some other or some graphical methods. So that depends upon your requirement, and then we can find out how many nodes or how many correlations are established with that protein, and then we can predict whether the, I mean, what mechanism might be working in that with that drug.
Yes. So, adding to this, if you are looking for the polypharmacological activities also in Biodiscovery Studio, we have a very unique feature called the ligand profiler, where you can have the multiple ligands molecules with the multiple macromolecules, where you can check whether that particular small molecule will interfering with the different pathways or with each and every macromolecule or not. So there you can study about the different polypharmacological activity to avoid the toxicological properties of that particular ligand molecule if it is interfering with the different pathways or not. So there are different ways of doing those kind of virtual screening of your small molecules to get that particular hit molecule actually. So on the basis of these bioinformatic tools, which make our work little easier to identify that particular hit molecule against a macromolecule, that's how they are helpful to us. Yes. So, any other questions? So, thank you so much, ma'am, for this insightful session. Now it's time to felicitate our speaker of the day with the memento and certification of appreciation. Thank you once again, ma'am, for giving your time and for such a great interaction with us and our viewers. It was pleasure to have you with us today. So stay tuned with us, guys. Please subscribe, sir, like us, and follow us for upcoming events. Let us connect and grow together. Thank you. Thank you all. Thank you so much, ma'am.
Thank you.